Hi everyone, my name is Philipp. I head up the Bioinformatics in the applications team at Oxford Nanopore. So if you've heard these days, in the applications team, we work on quite a few different areas. But at its core, I would say what we try to do is to demonstrate how to use the advantages of nanopore sequencing and how to apply them to specific real-world use cases. So why am I telling you this? Basically just to motivate my goal for today. So I'm not a geneticist, I'm not a researcher, so my goal is not really to show you any new interesting biological insights. There's a long list of amazing talks in these 2 days who do that way better than I ever could. Instead, my goal for today is to show you a few different ways of how to use nanopore sequencing and hopefully give you a few ideas how nanopore sequencing could assist you in your area of research. And for for today, we chose the topic of basically kind of like a multi-omics exploration of the disease mechanisms that underpin a rare disease that's called Rett syndrome. And with this example, I hope I'll be able to show you why basically just having a variant call is not always sufficient. But let's start at the beginning. What is Rett syndrome? It's a rare genetic disorder, as I said, that affects brain development. And in the vast majority of cases, it's caused by de novo mutations in a gene on chromosome X called MECP2. And MECP2 is this kind of really fascinating, amazing gene that does loads of different things, and we could have a whole talk about exploring that, but today we're focusing a bit more on the disease mechanisms behind Rett syndrome. More importantly, I should probably clarify a bit what I mean when I say multi-omics. So that term can obviously mean different things, but what I'm going to talk about today is how you can use genomics data, so genetic variation calling, and integrate and combine that with epigenetic signals like methylation or chromatin accessibility, and then combine that or kind of use transcriptomics data to confirm findings from those two. Right, so why would you use Oxford Nanopore data for that? Kind of the very simple answer is that even with a single data set, there's actually a lot of information you can get. So even if we just look at whole genome sequencing, you basically have 3 different channels of information. Obviously, you'll get canonical bases, and with those, with long reads, they will allow you to basically explore the full spectrum of genetic variation. And also, not only that, you can not only call them, you can also phase those variants, meaning you can figure out which of these variants occur on the same copy of a chromosome. In addition, as everybody of you knows, you can read out native modifications like 5MC and 5HMC. So they're important for various things like imprinting disorders or repeat expansions or methylation signatures. But then the third one is really interesting. It's something that more and more people are exploring, and that is using synthetically introduced modifications that otherwise don't really occur in humans, for example 6MA, to basically paint additional information on your reads. And so there are different protocols to do that. One example what people often do nowadays is basically paint information about open chromatin on the reads using 6MA. There are different protocols, for example, SMAC-seq or FiberSeq, which do that. And so kind of the screenshot that I'm showing you here gives you one example. And kind of like very naively speaking from a bioinformatician, you introduce a methyltransferase into the nuclei, and basically all the regions that are accessible will be methylated by that methyltransferase, whereas the ones that are protected won't. And that's the signal that we read out. So that's what you see, for example, on this screenshot here, where like all the regions that have like are purplish, that indicates that there's open chromatin there. Another thing you can do with this, for example, if you're interested in protein-DNA interactions, or something like a ChIP-seq experiment, there's the DAMELO-seq protocol that will do that for you. But today, for this talk, we'll focus on the open chromatin. One thing to point out here, and to come back to the initial question of that slide, is that this screenshot is from the same region in the same data set. And the only thing you need to do to get from one to the other is basically change your color by option in your genome browser. So this is kind of like the amount of information you get just from whole genome sequencing. So for this project, what we did is we got 9 different Rett syndrome cell lines. So these are not patient samples, they're just research samples. We did whole genome sequencing for those 9, again, using 6MA to mark open chromatin. Then for 3 more of those, we also got POR-C data, which is Nanopore's version of a chromatin conformation capture. And then for 6 more of those, we also sequenced 40 to 50 million long cDNA reads. Obviously, it's a lot of kind of data to keep track of, so I'll try on all slides that show any kind of results, I'll try to mark and highlight which data sets were used, and that's what you see kind of on the bottom left here. LSK is the whole genome data, cDNA, obviously cDNA, pore C is pore C. Right. As I said initially, the vast majority of Rett syndrome cases are caused by de novo mutations in the MECP2 gene. And interestingly, there's a wide range of different variants that can affect MECP2. So for me, it's a perfect use case to kind of demonstrate our human variation workflow to you. End-to-end workflows are currently a big focus at Nanopore, and the human variation workflow is a good example. It basically starts from DNA extraction and goes down to analysis or interpretation. On the next slide, I'll just briefly touch on how that looks for MECP2, but if you're interested in the whole process, we have a poster on it as well. Right, so as I said, there's kind of like various different variants. The first class is kind of missense SNPs. They typically occur in the binding domains of MECP2, and they're generally considered to be less severe. Here I show you one example that's called using our epitomy workflow, and then kind of prioritized as likely pathogenic using one of the now kind of tightly integrated tertiary analysis providers that support ONT data. The next class is frameshift mutations. Unsurprisingly, they're generally more severe. Here I'm showing you an example of a one base pair deletion. And then last but not least, there's a wide variety of a larger structure variation that can affect MECP2, like for example this 8 kb deletion that removes exon 3 and parts of exon 4 and was also marked as pathogenic by tertiary analysis. So these are really variants that we expected to find in these samples. After all, it's well-characterized cell lines. What we didn't really expect, but what was highlighted by the epitomy report, is a repeat expansion in RFC1. So repeat expansions, like biallelic repeat expansions in these specific genes, are implicated in a neurodegenerative disease called canvas. And so what we see here, what I'm showing you here, is two different plots from the epitomy workflow, which just basically shows you the repeat length distribution. And you can see that both samples have one heavily expanded allele, and the left one has a not expanded allele, and the right one a moderately expanded allele. The interesting thing about this repeat expansion is that it's not only about the length, but it's about the motif. And so since we have long reads that basically just span the whole repeat, we can just go down and look at these motifs. And when we do that, we see that in both samples, the heavily expanded allele actually consists of the most well-known pathogenic motif. Whereas on the left, the short one is the most well-known non-pathogenic motif, and then on the right, the moderately expressed one, kind of like expanded one, is a less well-known motif, but it's also believed to be non-pathogenic. So kind of in summary, like none of them would be considered having a biallelic pathogenic expansion, but nevertheless, I found this to be a kind of like a very interesting example to show what you can do with nanopore data in repeat expansion diseases. So I hope this kind of like gave you a quick overview of what you can do, how you can cover like the full spectrum of genetic variation with nanopore and the end-to-end human variation workflow. One thing that's really important with long reads, so you can't, it's not that you can just call the variants, you can also phase them, meaning you can figure out whether they're on the same copy of a chromosome or not. And so with the data that we have here, there's a couple of ways that we can do it. We'll start with the most kind of like straightforward one, kind of single sample, single data set. You just use the information that you get from the long-read whole genome sequencing data, and you phase the variants and then the reads. And what you will find with a typical ONT data set is with this, you get kind of like continuously phased phase blocks of roughly megabase size. And then with this, you'll see that roughly 70 to 80% of your genes will be fully contained in the phase block, so be fully phased. And that's very interesting and super helpful if you look for example, compound heterozygous variants. And like, while in Rett syndrome, compound heterozygous variants are actually not relevant, I'll show you another example today where it actually is. One limitation that you have with this kind of phasing is, like within a phase block, obviously, the phasing is consistent. But for the phase blocks, you don't know whether haplotype 1 or haplotype 2 is paternal or maternal. That's just something you don't get. And also, you can't make any assumptions of variants that are not in the same phase block. If, for whatever reason, you need that, you can go to kind of like more traditional ways of doing it, which is trio or dual sequencing, which will allow you to, first of all, in the offspring, get parent of origin assignment, and then also get basically fully phased chromosomes. One slightly less well-known option of doing it, when you actually don't have access to parental data, is you can throw in poor C as well. If you put in poor C on top of the long reads, you will also get basically fully phased chromosomes. And later in my talk, I'll show you an example where this is useful. But for now, we'll just focus on long reads alone. So why am I telling you about phasing? Basically, I'm just trying to build up to come back to the thing that I said in the beginning of sometimes a variant call alone just isn't sufficient. And that's perfectly illustrated by this family here. So here there was actually no cell line available for the father, but we have data for the mother and for the two offspring, a daughter and a son. And interestingly, while the daughter and the son are affected by Rett syndrome, the mother isn't, although they all have the same pathogenic variant. Again, we ran it through the workflow. It's annotated with tertiary analysis. All three have the same pathogenic variant, but only the children are affected and the mother isn't. So the question is, like, what is happening here? How does that work? The first thing we wanted to look at was, are there any kind of expression differences between the haplotypes in these three samples? So with the data that we really have two options to do that. We can, if we just look at our whole genome sequencing data, we can use the open chromatin information as a proxy for expression. So just basically check, is there open chromatin in the promoter region of MECP2, which again would be a proxy for it's expressed or not expressed. And then we also have cDNA data, so we can actually just directly check whether it's expressed or not. But first, let's focus on only having the whole genome data. What we need to do there, though, and this is where phasing comes into play. So this is data from the mother. This is the pathogenic variants at the bottom here. And what we need to figure out is whether the promoter, the MECP2 promoter, on that allele is in open chromatin or not. And for that, we need to phase basically the end of the gene, like this is the last exon of MECP2, with the promoter that's upstream of MECP2. And in this example, that's 80 kb. But with long-read phasing, we can just do that. And if we do that, we see that actually only the top track, the haplotype without the allele, has open chromatin, whereas in the bottom, the haplotype with the affected allele doesn't have any open chromatin. So just from this, just from looking at the whole genome data, our expectation would be the top one is expressed and the bottom one isn't. As I said, we have cDNA data, so we can just go and check. And when we did that, we confirmed that basically there is not a single read, not a single transcript that shows the pathogenic variant. So it's completely silenced. If we do the same analysis for the daughter and the son, we see that in the daughter in the middle, you have open chromatin on both haplotypes. So we'd expect that both haplotypes are expressed, and we see that in the cDNA data, roughly 13% of the transcript have the variant. In the son, there's only one copy of the X chromosome, so all the reads have the variant. So this kind of like explains why you can have the mother with the same pathogenic variant that's unaffected while the kids are affected. But the question is still, what's the mechanism behind that? And basically, everybody who was in the human clinical research session today will know already that the answer is X chromosome inactivation. So in any given cell, only one copy of the X chromosome is expressed. In males, there's only one copy, so that's it. In females, we have two. One needs to be deactivated. And what I didn't appreciate fully before starting this project is how this actually works. So apparently, kind of early on in embryonic development, each cell just randomly chooses one for deactivation. And then this kind of like, this decision is kind of kept throughout subsequent cell divisions, and that means kind of like in the individual, you would expect 50% of the cells choose one, 50% chose the other. So 50% of the cells will express the paternal X chromosome and the other 50% the maternal, because the assumption is that that process is random. So this is what the top one, this is what we would call basically a non-skewed X inactivation. However, for various different reasons, for example, having a deleterious variant on one of the chromosomes, this could be skewed. It could be 20/80, 90/10, or even 100/0. So obviously, kind of like you can see from the family, from the example that I showed you, that this is relevant information if you talk about disease or disease severity. And so the question here obviously is kind of, can we quantify it, can we detect it? And the key thing here is that same as in genetic imprinting, the deactivated locus is heavily methylated. And that's a signal we can read out. So just to give you a visual representation of that, I put one example of a promoter that is imprinted. It's from the Prader-Willi locus. and then next to it, one promoter on the X chromosome. And in imprinting, the kind of like deactivation is parent of origin specific. So you will see something like here, where like one haplotype is fully methylated, the other one is fully unmethylated. Whereas in the inactivation case, this is actually kind of like a non-skewed case. You see that in each haplotype, roughly 50% of the reads are red, the other 50 are blue. So 50 are methylated, 50 are unmethylated. And this also gives us kind of intuitively a way of quantifying this, right? If you have a heavily skewed X inactivation, it will look more like the imprinting case. So one thing that we can easily do is we can just get the methylation frequency separately for the haplotypes. For the inactivation here, it would be 50-50. For the other one, it would be 0 and 100. And then by comparing those, we can quantify X inactivation. So this is just one locus. We basically did the same thing for the whole chromosome. And this is what I'm showing you here. Basically, on the x-axis, there is kind of positions on the X chromosome. On the y-axis is the log 10 ratio of the methylation from the two haplotypes. So if it was 50-50, here it would be 0. If it's 0 and 100 or 100, 0, it would be 2, +2 or -2. I'm just showing you an example from a healthy control. There's a bit of skewing, but not that much really. It's kind of expected, it's moderate. In the daughter, if we look at that, it's slightly more skewed, and then in the mother, you see it's heavily skewed. So this is kind of completely consistent with what we have seen. So this is the mechanism how one haplotype in the mother is completely silenced, whereas in the daughter, you have 30% of the transcripts having that variant. One thing that you might kind of like see here is that those bars don't go in the same direction. Some go up, some go down. And this comes back, and it's basically one of the reasons why I showed you this slide earlier, because so far we have used long-read sequencing. So within the phase block, we can assess the skewing, but in the next phase block, we don't know if haplotype 1 and haplotype 2 are the same or switched around, which is perfectly fine if you just want to assess X skewing, because we expect it to be the same across the chromosome. So it's not a problem, but in the last couple of slides, I just want to give you an example where you can see what you can do if you throw in trio or, in this case, poor C phasing to get fully phased chromosomes. If you do that and basically plot exactly the same thing, this is how it looks. And now it's, I think, looking more like you would expect it, so the skewing is consistent. It's always towards one haplotype. But then there are these kind of like tiny outliers here. And you could think, well, it's artifacts, it's errors, or something like that. And some of them might be, but some of them actually indicate another disease-relevant mechanism that is part of X inactivation, which is that some of those genes escape X inactivation. So apparently it's known that roughly 50% of the genes escape X inactivation, so are expressed on the silenced allele. And so what we want to do is we basically want to figure out, can we identify them for our samples? And again, can we identify them only using genomic data and then also using cDNA data? So what we did is we took our two fully phased haplotypes. We know that haplotype 1 is the silenced one. We looked at all the regulatory elements annotated on chromosome X, and we just checked, are they in open chromatin or not? So again, we used that as a proxy of, are they escaping or not? And what we found is that the vast majority of the elements are kind of in open chromatin on haplotype 2, and only a small proportion is in open chromatin haplotype 1. And so this is kind of like, if you look at the numbers, this is what we see. And if you divide this basically by the total number of elements on chromosome X, we see that also in our data, we get roughly 15 to 20% of the genes escaping inactivation in kind of like the silenced allele. And just to go a bit further in validating that, we looked at the literature and basically a list of really well-known genes that typically escape inactivation, and we consistently found that they agreed with our results. So again, there's no new biology here, but it's actually kind of mind-blowing how simple it is to find these on the whole chromosome, fully phased. So that was kind of like a very nice finding. And then on my very last slide, I just want to show you again, we have cDNA data for this, so we can go in and we can even verify this and see if this kind of like open chromatin proxy works in some cases or not. And so what we have to do for that is we have to look at allele-specific expression along chromosome X. And so long cDNA reads really help, because for that we basically only need one heterozygous variant in the coding region to do that. And this is an example where I'm showing you this. Basically here you have this one SNP in the first exon, And that SNP allows us to phase the reads and assess allele-specific expression. So this shows us that indeed both haplotypes, both copies of these genes are expressed, and so the gene escapes X inactivation in this sample. And again, just to point it out, it nicely aligns with our open chromatin signal in the promoter. And if we kind of try to assess how much of the expression stems from the inactivated allele, we roughly get 35%, and that also nicely matches up, which is known from what's known in the literature. So that was a really nice confirmation as well. And with this, I want to kind of like end my presentation and just summarize that. I hope I was able to show you how you can use nanopore data to, again, look at the full spectrum of genetic variation, then how you can use phased modification to look at disease-relevant mechanisms like imprinting or X inactivation, how you can use synthetic modifications to add additional information to your experiment like open chromatin, and then kind of just hint at how you can use transcriptomic data to kind of like either combine it with the former or confirm findings from there. And the last two things I will say were, the last two things, I hope I was also able to show you how kind of like phasing pulls all of this together and allows you to do all these things in a haplotype resolved fashion. And now the last thing I'm going to say is that we're really just scratching the surface here. MECP2 is a fascinating gene. It does loads of things. It binds 5MC, 5HMC, and kind of like modulates transcription and affects kind of like chromatin accessibility on a large scale and on a small scale. So there's loads to explore, and we could explore this with this data set, but that might be a talk for London Calling. So with that, thank you very much for your attention.